Chapter 25 Glycogen Synthesis. Let's look at the big picture again. This chapter is about from glucose synthesized to glycogen. Carbohydrate-rich meals are consumed before an endurance sports event to ensure the presence of the muscle glycogen required for the sport. This chapter we're going to study glycogen is synthesized and degraded by different pathways, and glycogen breakdown and synthesis are reciprocally regulated. Let's look at the glycogen synthesis and degradation compared to two different pathways. In glycogen degradation, the glycogen undergoes phosphorylases, release glucose in the form of glucose-1-phosphate. While in the synthesis, the glycogen receives glucose from a UDP glucose. This UDP glucose is an activated precursor and glucose donor. It has the structure of this is a uridine diphosphate glucose. The separate pathway of glycogen synthesis and degradation afford much greater flexibility, both in energetics and in control. The glycogen degradation yields glucose-1-phosphate, while the glycogen synthesis, the glucose is added through the UDP glucose. UDP glucose is a monomer that used to extend the glycogen chain in synthesis. A little bit of review. The PI is orthophosphate. ATP is active carrier of PI. Just like UDP glucose is active carrier of glucose. Let's look into UDP glucose. UDP glucose is a activated form of glucose, a glucose donor, or say glucose carrier in glycogen synthesis. It's synthesized by UDP glucose pyrophosphorylase. The reaction is irreversible after the hydrolysis of a pyrophosphate. Here's the equation. Glucose-1-phosphate react with UTP, form an equilibrium of UDP glucose plus pyrophosphate. And then when the pyrophosphate hydrolyzed with water, the reaction becomes irreversible. Glucose-1-phosphate turn to UDP glucose. Glycogen synthesis catalyzes the transfer of glucose from UDP glucose to a growing chain. The glycogen synthase, a key regulatory enzyme in glycogen synthesis, transfer a glucose moiety from UDP glucose to the C4 terminal residue of a glycogen chain to form an alpha-1,4 glycosidic bond. Glycogen synthase requires a oligosaccharide of glucose residue as a primer. The primer is synthesized by glycogenin, a dimer of two identical subunits. Each subunit of glycogen generates the oligosaccharide of glucose residues 10 to 20 glucosol units long. Glycogen synthase then extends the primer. In another word, the glycogen starts from a core protein, the glucogenin. Glucogenin generates a short chain of glucose residues about 10 to 20 glucosol units long, and this short chain acts as a primer for the glycogen synthase to add glucose onto the short chain to extend it. And the location to add this glucose is at the C4, and the bond form is alpha-1,4 glycosidic bond. We're going to introduce another enzyme called branching enzyme. It forms alpha-1,6 linkage. The glycogen synthase can only synthesize alpha-1,4 linkage. It starts from the primer. The glycogen synthase transfer glucose from UDP glucose onto this primer chain, extend the chain. Then a branching enzyme cleaves a alpha-1,4 linkage and taking a block of about 7 glucose and synthesize a alpha-1,6 linkage. The glycogen synthase then extend the branched polymer. Synthase extends both non-reducing ends, followed by more branching. Glycogen synthase is the key regulatory enzyme in glycogen synthesis. Glycogen synthase exists in two forms: an active non-phosphorylated A form and usually inactive phosphorylated B form. The conversion of the two forms is regulated by covalent modification. The key regulatory process for glycogen synthase is the conversion of the B form from inactive T state to active R state by bonding glucose 6-phosphate. Note that the phosphorylation has opposite effect on glycogen synthase than on glycogen phosphorylase. Both enzymes have two forms, A form and B form, but the phosphorylation is different. It's opposite. And also the regulatory process are different. For the glycogen synthase is to conversion of the B form from T state to R state, while the glycogen phosphorylase is to convert from inactive B form to active A form. Do you know that study shows that when muscle glycogen is depleted, the power output of the muscle falls approximately 50% of the maximum. The power output decreases even though the body have enough fat available, suggesting that fats can supply only about 50% of maximal aerobic effect. If carbohydrate-rich meals are consumed after glycogen depletion, the glycogen stores are rapidly restored. In fact, glycogen synthesis continues increasing glycogen stores far above normal. This phenomenon is called supercompensation, or more commonly called carbo loading. This explains why if you want to limit your carbohydrate intake, the calorie intake, and the weight, you can lose weight initially, but eventually it most likely rebound. The weight will rebound. Glycogen is an efficient storage form of glucose. Only 2 molecules of ATP are required to incorporate dietary glucose into glycogen. The complete oxidation of glucose derived from glycogen yields 31 molecules of ATP. Let's look at the synthesis. Glucose needs 1 ATP to form glucose 6-phosphate. And the glucose 6-phosphate will convert it to glucose 1-phosphate and turn to UDP glucose and eventually add a glucose onto the glycogen chain. And in summary, the reaction is glucose need 2 ATP to add it to a glycogen chain. For breakdown, 88% of residues are cleaved by phosphorylases to gluco-1-phosphate, which is converted into glucose-6-phosphate without expanding actual ATP molecule. And 12% are branch residues that can be cleaved to free glucose, and one molecule of ATP is needed to turn to glucose-6-phosphate. So in overall, 2 ATP are required for storage in glycogen form, but glycogen-derived glucose generates 31 molecules of ATP when complete oxidation of glucose-6-phosphate. So the overall efficiency of storage is nearly 90%. Glycogen breakdown and synthesis are reciprocally regulated. The glycogen synthesis is inhibited by the same glucagon and epinephrine signaling pathways that stimulate glycogen breakdown. Phosphorylation of a glycogen synthase by protein kinase A to form glycogen synthase B inhibits the glycogen synthesis. Glycogen synthase kinase also phosphorylate and inhibit glycogen synthase. This is a diagram of coordinated control of glycogen metabolism. During exercise or fasting, the glucagon or epinephrine signal the cell to trigger a cascade of reaction. The ATP will use to synthesize cyclic AMP and activate protein kinase A, active phosphorylase kinase, and phosphorylase, in that active the glycogen breakdown. When this pathway is dominated, when protein kinase A is active, the protein kinase A and the glycogen synthase kinase, they inhibit glycogen synthesis by convert glycogen synthase A to glycogen synthase B. The B is inactive form. From last slide, we learned that the kinase inhibit glycogen synthesis. Protein phosphatase 1 reverses the regulatory effect of kinases on glycogen metabolism. Protein phosphatase 1, PP1, shifts glycogen metabolism from the degradation mode to the synthesis mode. PP1 removes phosphoryl group from glycogen synthase B, converting it into the more active A form. And PP1 also removes phosphorus group from phosphoryl kinase, and phosphorus A inhibit the glycogen degradation. Let's look at the chart. After a meal or when at rest, when glucose level is high and is at the high energy charge, the protein phosphatase 1 would remove a phosphoryl group to inhibit the phosphorylase kinase, and also can dephosphorylase a phosphorylase A to phosphorylase B, and also can dephosphorylase a glycogen synthase B to convert it to glycogen synthase A, which stimulate turn on the glycogen synthase. Let's look at the protein phosphatase 1 structure. It consists of a catalytic subunit PP1, marked blue color, and one of a family of regulatory subunits, orange color. A key regulatory subunit is the G subunit, GL in liver and GM in muscle, that binds glycogen and the catalytic subunit PP1, localizing the enzyme with its subunits. In muscle, the phosphorylation of GM results in the release of a catalytic subunit of PP1 and a decrease in the enzyme's activity. Almost all tissue contains small proteins that, when phosphorylated, inhibit the catalytic subunit of PP1. From the chart, we can see phosphorylation of GM by protein kinase A dissociates the catalytic subunit from its substrate in the glycogen particle. Phosphorylation of the inhibitor subunit by protein kinase A inactivates the catalytic unit of PP1. Insulin stimulates glycogen synthesis by inactivating glycogen synthase kinase. Insulin stimulates glycogen synthesis by activating a signal transduction pathway that results in the phosphorylation and inactivation of glycogen synthase kinase. The PP1 subsequently dephosphorylate glycogen synthase, generating the active A form of the synthase. Insulin also facilitates glycogen synthesis by increasing the number of glucose transporter GLUT4 in the plasma membrane. You can see from the figure that the insulin bound with the insulin receptor and triggered a cascade of reaction active the protein kinase. The protein kinase phosphorylate the glycogen synthase kinase. The phosphorylation inactivated the glycogen synthase kinase, and therefore the glycogen synthase B is dephosphorylated by the PPI to be the glycogen synthase A active form. Glycogen metabolism in the liver regulates the blood glucose concentration. Insulin and high glucose concentration signal the glycogen synthesis in the liver. When glycogen synthesis is activated, glycogen breakdown is switched off. The liver senses the concentration of glucose in the blood. Glycogen phosphorylase A bonds the glucose and converts from R state to the T state. The conversion releases PP1, which is then free to activate glycogen synthase and inactivate glycogen phosphorylase. PPI converts glycogen metabolism from a degradation mode to a synthesis mode. The activity of glycogen synthase begins to increase only after most of the phosphorylase is inactivated. The lag between degradation and synthesis prevents the two pathways from operating simultaneously. In muscle, phosphorylase A is unaffected by glucose and is affected by the rise in blood glucose concentration. This diagram depicting glucose regulation of liver glycogen metabolism. Glucose regulates liver glycogen metabolism. In the liver, glucose bonds to a glycogen phosphorylase A and inhibits it, facilitating the formation of the T state of the phosphorylase A. The T state of phosphorylase A does not bind protein phosphorytase 1, PP1, leading to the dissociation of the PP1 from glycogen phosphorylase A and its subsequent activation. The free PP1 dephosphorylates glycogen phosphorylase A and glycogen synthase B, leading to the inactivation of glycogen breakdown and the activation of glycogen synthase. Clinical insight. Diabetes results from insulin insufficiency and glucagon excess. Diabetes is characterized by the presence of excess glucose and underutilization of the fuel. Excess glucose is secreted in the urine. In type 1 diabetes, insulin is not produced. In type 2 diabetes, insulin is produced, but the insulin signal pathway is not responsive, a condition referred to as insulin resistance. Glycogen storage diseases result from a variety of biochemical defects in glycogen metabolism. Here's a list of several diseases and its defective enzyme, organ affected, glycogen in the affected organ, and clinical features. We can see in Cori disease, the defective enzyme is alpha-1,6-glucosidase, the deep branching enzyme, and affected organ is muscle and liver. Increased amount, short outer branches, and the clinical features including like type 1 but milder course.